So dear students, now we are going to start our next chapter, that is inflammation. So next topic next chapter, the second chapter we are going to study is inflammation, right? So first of all, we should know that whether inflammation is harmful or helpful, right? So we have to understand that inflammation is both protective and damaging. So it protects also, it's a protective response from the body, and it's the damaging response from the body, both, right? So inflammation is both protective and damaging. Protective, they will be protecting against infections. Damaging, as you can see in autoimmune disorders, so that time it will be damaging. So just I'm giving you one example: protective against infections, and damaging example will be autoimmune disorders, right? So basically we divide inflammation into two broader categories: acute inflammation and chronic inflammation. So we divide inflammation into two categories. So here I'm writing inflammation, right? Inflammation can be of two types: acute and chronic. So what is the difference between acute and chronic inflammation, right? Acute and chronic inflammation. Acute inflammation usually of shorter duration. So when you will see, acute inflammation will be of shorter duration, right? So it will be having shorter duration, whereas chronic will be having longer duration, right? So they will be having longer duration. So shorter duration means minutes to hours, right? So it will be ranging from minutes to hours. So it will be ranging from minutes to hours, whereas it will be ranging from days to months, right? So that is how we devise, right? Acute versus chronic inflammation. Another important distinguishing feature is about the cell. Which cell is predominant in acute inflammation? In acute inflammation, neutrophil is the dominant cell. So that is another important thing. In acute inflammation, neutrophil is the dominant cell, and in chronic inflammation, cells are usually macrophage and lymphocytes. Macrophage and lymphocytes. So macrophage and lymphocytes are important in chronic inflammation. When you see the severity of the inflammation, so usually acute inflammation will be showing more clinical symptoms, right? So there will be more clinical symptoms and signs, right? So symptoms and signs will be more, and in chronic it will be milder. So these are some of the basic differences between acute and chronic inflammation, right? So we will discuss one by one. First we will start from the acute inflammation, right? So first we will study about acute inflammation. So already we have discussed shorter duration, neutrophil will be the dominant cell. So in acute inflammation, what are the events which will happen in our human body? So we are going to discuss in two headings, right? One is cellular events of acute inflammation, and one will be vascular events of acute inflammation. So we are going to discuss acute inflammation into two broader headings. One is vascular events. So vascular events of acute inflammation, right? This is the vascular events of acute inflammation, and then we will be discussing about cellular events of acute inflammation. So vascular and cellular events of acute inflammation. What I mean to say that what is happening inside the blood vessel, right? What is happening inside the blood vessel, that will be vascular event, and what is happening to the cells of acute inflammation, right? So here I will teach you about what is happening to the cells of acute inflammation. So how they are migrating to the outside extravascular environment. So this will be cellular events of acute inflammation. So this is how we are dividing acute inflammation into vascular versus cellular events. So first we will start from the vascular event of acute inflammation. So first I will talk about vascular event of acute inflammation. So what happens in vascular events of acute inflammation? So it is related to the blood vessel. So whenever any injurious stimuli is given to a blood vessel in acute inflammation, what will be the first sign? What will be the first change, right? This is how examiner will be asking. So that point we have to keep in mind. So I am going to demonstrate that thing in a diagrammatic way. Suppose I am showing you this is the blood vessel, right? Just a pictorial illustration only, right? This is the blood vessel which I am showing you here, right? And we all know that blood vessel is lined by endothelial lining. So these are the endothelium. Say these are the endothelium. So blood vessel is lined by endothelium. So whenever there is an injurious stimuli given to a cell, see this, what I am writing here, injurious stimuli, right? Because of this injurious stimuli, there is a acute inflammation. So examiner is asking what will be the first response. So first response, because of this, there will be transient vasoconstriction. So what I am showing you here is a transient vasoconstriction. So this is the first response, right? So I am writing here, first response will be vasoconstriction. Vasoconstriction. So narrowing of the blood vessel lumen is called as vasoconstriction. Narrowing of the blood vessel lumen is called as vasoconstriction, and we must remember this is the earliest response. Examiner asked this question, what is the earliest response in acute inflammation? So earliest response in acute inflammation is the vasoconstriction. So that is how we have to keep this in mind. Earliest response is vasoconstriction. After this vasoconstriction, what will be the next step in acute inflammation? Next step in acute inflammation will be, right? After that, transient vasoconstriction will be followed by vasodilation, right? So now I am showing you the second event. Second event, what we are going to see is the vasodilation. Vasodilation, right? So this is the vasodilation. When blood vessel will dilate, so what will happen? The endothelial gap, right? It will be increased, right? So now we can see that because of the vasodilation, endothelial gap has been increased. So this gap is increased in acute inflammation, right? So this is called vasodilation. Because of increase in gap, what will happen? Whatever substances and fluid are there inside the blood vessel, they will leak outside now. So through this gap, fluid will be coming outside, the blood will be coming outside, some of the protein will be coming outside. So as a result, what is happening? We are seeing that it is called as increased vascular permeability, because endothelial gap has been increased. You can see endothelial gap has been increased, so vascular permeability has been increased. So because of this vasodilation, what is the third event we are seeing in acute inflammation? There is a increased vascular permeability. Increased vascular permeability, right? So increased vascular permeability. So what we are observing, earliest was vasoconstriction followed by vasodilation, then there is an increased vascular permeability, and because of increased vascular permeability, what we are observing, the fluid is leaking outside. When fluid is leaking outside, so what will happen? RBC count will be increased within the lumen, right? Or you can say cell count will be increased within the lumen, and fluid is going outside. So assume that if it is happening like this, number of cell is more and fluid is less, so what will happen to the viscosity, and what will happen to the movement of the cells within the lumen, right? So viscosity will be increased and blood flow will be decreased because fluid is less. So that is how we can understand the fourth step in acute inflammation. So as a result, what we are going to notice here? Because of the increased vascular permeability, there is a loss of fluid. Because of loss of fluid, there will be increased viscosity, which is causing stasis, right? So because of loss of fluid, there will be stasis, right? So fluid movement will be decreased. Why there is a stasis? Because of increased viscosity and increase in cell count, right? So because of the loss of fluid, you can see viscosity was increased, cell count was increased, and that is why stasis has developed. So these are the 4 sequential steps of vascular event of acute inflammation. and this was All India question in 2015. So what is the sequence of vascular event in acute inflammation? That was the question, and your answer is vasoconstriction, then vasodilation, increased vascular permeability, and then stasis, right? Another important MCQ which examiner asked here, what is the hallmark of acute inflammation? So remember, increased vascular permeability is hallmark of acute inflammation, right? So this is the answer, right? So this was the another important MCQ. What is the hallmark of acute inflammation? So hallmark of acute inflammation. So hallmark of acute inflammation will be increased vascular permeability. So remember this point, increased vascular permeability is the hallmark of acute inflammation, right? So that was the another important MCQ which examiner had asked. So 4 sequences we have seen: vasoconstriction, vasodilation, vascular permeability increase, and stasis, right? Why stasis is necessary? That is another important thing. Stasis, actually it will connect the vascular event to the cellular events of acute inflammation, because of the slowness of the movement. When you will see the neutrophil or inflammatory cells, suppose I am showing you inflammatory cell, this is an inflammatory cell, and just assume that this is the area where the inciting bacteria is residing. So bacteria is here. Because of this bacterial stimulus, now neutrophil has to come outside to kill this bacteria. To kill this bacteria, if you look at the normal blood flow, it is following the central axis, right? Midline, in the midline. But when they have to kill the bacteria, they have to move outside and they have to come to the periphery. So to come to the periphery, when they will come to the periphery, when blood flow will be decreased, right? So that is why stasis will happen. So purpose of stasis is for migration of the inflammatory cell to the periphery. That is the important thing to understand. Purpose of stasis is for margination of the neutrophil to the periphery of the lumen of the blood vessel. So that is why stasis is important, right? Coming back to the vascular events, when we were seeing vascular permeability is hallmark of the acute inflammation. So we will discuss little detailed concept of vascular permeability, right? So now we are going to discuss detail about this topic, which is very, very important, and that is vascular permeability. Vascular permeability, right? So what are the mechanisms responsible for increasing vascular permeability? This is the hallmark, but we should also understand what are the various mechanisms responsible for increasing vascular permeability. So here we have to understand vascular permeability. What are the mechanisms of increasing vascular permeability, right? So mechanism of increasing vascular permeability, number one and the most important mechanism is the endothelial gap formation, right? So this is the number one, endothelial gap formation, and remember this is the most important mechanism. This is the most important mechanism for increasing vascular permeability, endothelial gap formation. So endothelial gap formation is the most important mechanism, and after that second important mechanism is the endothelial damage, right? This is the another important endothelial damage. This endothelial damage we can divide like this: direct endothelial damage and mild endothelial damage. So these are two different things which we have to understand, right? So endothelial damage, here I will tell you, we can divide them into two categories. It can be direct or it can be, so this endothelial damage can be direct endothelial damage, or this can be mild endothelial damage. So direct and mild endothelial damage, right? Third one is the leukocyte mediated damage. So third important mechanism is leukocyte mediated damage. So this is the third mechanism, which is leukocyte mediated damage. So these are the three basic important mechanisms for increasing vascular permeability. So one by one we will see about them. Basically, examiner asks question about their nature of the stimuli, whether it is a immediate and transient, or delayed or prolonged, or immediate or prolonged, right? So we have to understand this response of endothelial gap formation. When you will see endothelial gap formation, what are the important things we have to keep in mind? Endothelial gap formation, this occurs at the level of venules, right? So remember, endothelial gap formation occurs at the level of venules, important MCQ, right? But there are exceptions, so endothelial gap formation occurs at venules except for lungs, and in lungs this will happen at the level of capillaries. So that is important to understand, right? Endothelial gap formation will be at venules throughout the body except for the lungs, right? So this exception was the question in All India exam, except for the lungs, and in lungs it will be capillaries, right? So that is the important question which examiner had asked in All India exam. In lungs, endothelial gap formation will be at capillaries. Exact question I will tell you when we will discuss further, right? So endothelial gap formation which we are seeing here is a kind of immediate and transient response. So this vascular permeability increase caused by endothelial gap formation is the immediate and transient response. So that is also another important MCQ which examiner asks you, okay, what kind of response it is. So this is the immediate and transient response. Immediate and transient. So this is immediate and transient response. So endothelial gap formation is immediate and transient response, right? Similarly, examiner asks this question, okay, what about the endothelial damage, right? So when we are talking about endothelial damage, you can see I have given two segments, direct endothelial damage and mild endothelial damage. So both are different things. When we are talking about direct endothelial damage, direct endothelial damage will happen immediately on the endothelium. So immediately endothelium will be damaged. So that will be immediate. But after endothelial damage, cytokines will be released and they will recruit inflammatory cell. So it will be prolonged. So immediate, prolonged response. So always remember, increased vascular permeability by direct endothelial damage will be immediate and prolonged. So immediate and prolonged response. So this is another important MCQ which examiner asks. Direct endothelial damage will be immediate and prolonged response. Whereas mild endothelial damage, when we are talking about mild endothelial damage, what will happen? Mild endothelial damage was the question in exam with this heading, sunburns. So when we are seeing sunburns, what will be the response? Sunburns is an example of mild, right? So mild is not, see this, direct endothelial damage is immediate and prolonged. Immediately it will damage, but recruitment of inflammatory cells for causing more permeability will be delayed. So immediate and prolonged. But milder injury, it is happening slowly. So it is delayed, and obviously after the damage it will recruit inflammation same way. So delayed and prolonged. So be careful when examiner says mild endothelial damage, this will be considered as delayed and prolonged. So here I would like to tell you, whenever examiner is asking question, you have to be very careful, because these were the questions in earlier exams. Okay, direct endothelial damage is the immediate and prolonged response. There was a question like this also, mild endothelial damage in sunburn is example of delayed and prolonged. So we have to be very careful when examiner uses the word direct means immediate and prolonged. Mild endothelial damage will be delayed and prolonged, right? Direct will be immediate and prolonged, mild will be delayed and prolonged. Leukocyte mediated injury, here we have to understand that leukocyte will come late and they will persist for longer time to produce increased vascular permeability. So this will be also delayed and prolonged. So this will be also delayed and prolonged response. So these are the important point about vascular permeability. So I will just sum it up. You can see for vascular permeability mechanism, as I said you, vascular permeability mechanism, endothelial gap formation, endothelial damage, then leukocyte mediated. So these are the three important one, and out of these three important one, what we have learned that endothelial gap formation is the most important mechanism which happens at the level of venules throughout the body, except for the lungs where it will happen in capillaries, right? So these are the vascular event of acute inflammation, right? Now we are going to see what is the cellular event of acute inflammation. So now we are going to move to the next topic, that is the cellular events of acute inflammation. So now we are going to discuss about cellular events of acute inflammation. So what will happen in cellular events of acute inflammation? So as I have told you, in cellular events of acute inflammation, it is the movement of the inflammatory cells towards outside, means towards the bacteria who is residing outside the lumen of the blood vessel, right? So now we will see what are the steps in cellular events of acute inflammation. Suppose I am drawing, this is the blood vessel. So remember, this diagram which I am going to draw is the schematic diagram for understanding point of view, right? So suppose I am showing you a blood vessel. As we have learned that blood vessel is having endothelial lining. So these are endothelium, right? We all know that in acute inflammation neutrophil will be there, and neutrophil is running in midline stream, right? So it is running in midline of the blood vessel lumen. Before I discuss about the cellular events of acute inflammation, I would like to tell you a simple brief introduction about the neutrophil, right? Brief introduction about the neutrophil. That is, in normal condition, neutrophil will be having two adhesion molecule on their surface, right? Remember, two adhesion molecules on their surface. One will be integrin, right? Integrin, here onwards I will be writing with this short form as I. So this is the integrin, and the second molecule what they will be having is called as sialyl Lewis ligands, right? So in short form I will be writing SL, right? So sialyl Lewis ligand I will be writing SL. So this is the two important adhesion molecules present over the surface of inflammatory cell, integrin and sialyl Lewis. When a neutrophil is under normal situation, integrin will be having low affinity. That is very important to understand. This integrin will be having low affinity, right? Integrin will be having low affinity. And when this inflammation will be going on, then this neutrophil integrin will be converted into high affinity integrin, right? So that is the basic concept we have to understand before we proceed to the acute inflammatory event or cellular events of acute inflammation. So now we can see that neutrophil was coming inside the lumen. They were running in midline, but because of the vascular event, due to stasis, they will be migrating to the periphery, right? So now we can see that neutrophil is migrating to the periphery. So it is coming to the peripheral area. So when they are coming to the peripheral area, you can see there is another endothelium which I am drawing here. There is another endothelium, right? This is the endothelium. This is the endothelium. And during inflammation, right, when this neutrophil is coming towards the endothelium, here integrin is under low affinity. So I am just writing downward arrow. Downward arrow means low affinity. So normal neutrophil was having low affinity. So when they are coming down and they are closing near to the endothelium, means they are coming to the periphery, still this integrin is inactive, means low affinity. But sialyl Lewis ligand, is there, which will combine with the another adhesion molecule which is called as E-selectin, right? So this will combine with E-selectin present on the endothelium. So during inflammation, endothelium will express E-selectin adhesion molecule, right? This E-selectin adhesion molecule will combine with sialyl-Lewis ligand. So this is how sialyl-Lewis will combine with E-selectin addition molecule, and there will be transient attachment of the neutrophil, or you can say weak adhesion of the neutrophil to the endothelial wall, and this is called as margination, right? So this is the number one event within the blood vessel called as margination. So remember, what is this number one event? Number one event is called as margination, right? So this was the question also in AIIMS exam. That is why I drew this diagram, so that we can understand. Question was, margination requires selectin addition molecule, true or false? So now you can see that yes, margination requires addition with E-selectin to the sialyl-Lewis, right? So what is margination? Transient. So see how I am going to define this margination. It's a transient attachment. It is transient attachment of neutrophil to endothelium. So transient attachment, so it's a weaker attachment. That is why I have written word transient. See why I have written word transient? Because it's a weaker attachment. So it's a weaker attachment of neutrophil to the endothelium. How? Via which addition molecule? Sialyl-Lewis and E-selectin. So via sialyl-Lewis ligand and E-selectin addition molecule, right? So that is the important addition molecule, sialyl-Lewis and E-selectin, and that was the question in AIIMS exam, right? E-selectin is required for margination. So that was the question. E-selectin is required for margination. So what is margination? It's a weaker and transient attachment of the neutrophil to the endothelium, so that a neutrophil can be attached when they are coming to the periphery of the blood vessel, so that they can sustain, or they can have a location at the peripheral area of the blood vessel, so that they can migrate towards bacteria later on in the cellular events of acute inflammation. So this is number one. After this, after margination, what will happen? Now again, I'll just extend this blood vessel and we will see. I'm again extending this blood vessel, and suppose this is the endothelium. This is the endothelium, right? Endothelium will have expression of another addition molecule, which is called as P-selectin. So there is another addition molecule which is called as P-selectin, right? This P-selectin, again this P-selectin will combine with the neutrophil. Now P-selectin will combine with neutrophil sialyl-Lewis ligand, right? So this sialyl-Lewis will combine with the P-selectin addition molecule, right? But remember, this P-selectin addition molecule will be giving transient attachment. So this transient attachment will be later on replaced with the E-selectin addition molecule. So that is how this transient attachment will be given by P-selectin, right? So this transient attachment and detachment, detachment. So like that it will be going on, and this is called rolling. So this neutrophil will start rotating because of the transient attachment to the P-selectin, then again E-selectin, then P-selectin, E-selectin. Like this it will be going on, and this will create tumbling movement of the neutrophil, which is known as rolling, right? So this is called, so P-selectin is required for rolling. So this is another important thing which examiner asks in exam, right? Okay, what is the function of P-selectin? So always remember, P-selectin is for rolling only. Rolling only, nothing else. P-selectin, remember, P-selectin is for rolling only. So that is another important older All India question, right? So P-selectin is for rolling only. So it is the tumbling movement which is because of transient attachment and detachment of neutrophil, right? So transient attachment and detachment of neutrophil to the endothelium with the help of P-selectin molecule will be responsible for rolling. So always remember, P-selectin required for rolling only. That is how examiner had asked question in All India exam, right? So P-selectin is for rolling only. This will provide transient attachment and detachment of the neutrophil to the endothelium like this, and now neutrophil will start rotating, right? So there is a tumbling moment. So after that what you will see, Now they have started rolling. So when this neutrophil will start rolling, what will happen? On the neutrophil surface, integrin will be activated. So now integrin has been converted into high affinity integrin. This is called high affinity. So now this is high affinity integrin, right? So this integrin is having high affinity, and Shelley-Lewis has been moved out, right? So after rolling, when integrin has been activated, this integrin will attach to the ICAM, intercellular adhesion molecule, or VCAM, vascular cell adhesion molecule, expressed on endothelium, again on endothelium, right? So ICAM or VCAM, these are the adhesion molecule present on endothelium. So ICAM, VCAM, intercellular adhesion molecule. ICAM stands for intercellular adhesion molecule, VCAM stands for vascular cell adhesion molecule. They are expressed on endothelium, and now this will give a strong attachment to the neutrophil. So see, what is the difference between margination? Margination, Shelley-Lewis E-selectin was the weaker attachment, transient and weaker attachment. But in this case, what we are going to see now, this is called adhesion, which is a stronger attachment. So this is the third step in the cellular event of acute inflammation. Third step is the adhesion. What is adhesion? So as I said, adhesion is the strong attachment. Adhesion is the strong attachment or firm attachment or adhesion. Strong attachment of neutrophil to endothelium, right? How? So it is via which adhesion molecule? So it is via integrin, right? So remember, high affinity integrin is combining with intercellular adhesion molecule or vascular cell adhesion molecule. So this is how it will help in a strong adhesion, right? And that was the question in AIIMS exam, right? What is the function of integrin? Integrin is for adhesion, right? So integrin is for adhesion. Neutrophil will be adhered to the endothelium with the help of integrin, which will attach to the ICAM or VCAM expressed on endothelial surface. This is called adhesion, right? So during this process of inflammation, what you are observing, so again I am extending this blood vessel. What we are observing on the endothelial lining, now we can see layer of neutrophil is present, right? So layers of neutrophil is present. So on this blood vessel, when you see layers of neutrophil, we are seeing layers of neutrophil over the endothelial lining. This is called pigmenting. So this is the fourth step. Layering or layers of neutrophil. This is the fourth step called as pigmenting. So what is pigmenting? Layer of neutrophil lying on the endothelial lining, right? So this is called overlayering. I will write overlayering of neutrophil on endothelial lining. So this is called as pigmenting, right? So this is called as pigmenting. So these are the 4 sequential steps during acute inflammation, which was asked in previous year AIIMS exam also. What is the sequence? So in this lumen, what sequence we have seen so far? It's a mnemonic which we can keep in mind to remember. There is a Michael Jackson who used to sing rap song, M rap. So these are the sequential steps we have to keep in mind, right? Sequential step is margination, rolling, adhesion, pigmenting, right? So this is how we can remember this by mnemonic, right? Margination, rolling, adhesion and pigmenting, right? So now we can see pigmenting is overlayering of neutrophil on the endothelial lining. So as we are discussing about acute inflammation or inflammation, during inflammation there will be production of the cytokine within the lumen of the blood vessel, right, or in body. So what I am showing you here, because of inflammation, see the concept which I am going to explain now. During inflammation, inside the blood vessel, cytokines will be synthesized from the endothelial lining, from the inflammatory cells and from the other cells also. So that is the point I wanted to tell you. During inflammation, cytokine will be synthesized. And who will be synthesizing? As I said, it can be any inflammatory cell, it can be endothelial cell, or it can be any other cell which are present in the blood vessel lumen or in our human body, right? So during inflammation, when cytokine is synthesized, what is the role of this cytokine, right? So what is the role of cytokine? Now we can see this cytokine will be having activating function. They will activate endothelium, right? So they will activate endothelium. When endothelium is activated, they will express adhesion molecule which is called as PCAM1, platelet endothelial cell adhesion molecule 1, right? So this cytokine will activate endothelium and endothelium will express PCAM1 adhesion molecule, right? The same cytokine will activate neutrophil also, and neutrophil will also express the same PCAM1, platelet endothelial cell adhesion molecule, right? So this adhesion molecule is expressed on endothelium also, on neutrophil also, right? Try to remember, try to understand. PCAM1 is an adhesion molecule which is expressed on both endothelium and neutrophil, and who is the reason? Reason is the cytokine. So cytokine is inducing PCAM1 on both neutrophil and endothelium. So what is the role of this PCAM1 adhesion molecule? This will help to attach neutrophil to the endothelium, and now this neutrophil will try to squeeze out between these two endothelial gap. So see, this is the endothelial gap, and we know that there is a basement membrane. So just I am making a simple diagram to show that this area is the basement membrane, right? So what I'm showing you here, this area is the basement membrane, right? So there is a basement membrane, there is an endothelium, and there is a gap between the endothelium. So neutrophil will try to squeeze through this gap with the help of PCAM molecule. So this process where I am showing you कि neutrophil is trying to squeeze through the endothelial gap is known as diapedesis, right? This is called diapedesis. Remember why I have drawn this basement membrane? So that we can understand diapedesis definition is transmigration of leukocyte between the endothelial gap, not through the basement membrane, not through the basement membrane. So that is the strict definition. So here I would like to tell you the fifth event is called as diapedesis, right? So this is the diapedesis. What is diapedesis? As I said, diapedesis is transmigration of the leukocytes. You can see transmigration of the leukocytes via endothelial gap. So this is how we will define diapedesis is the transmigration of leukocytes via endothelial gap. So that is important to understand what I am highlighting here, via endothelial gap, because that was the question in All India exam. What is diapedesis, right? So the question is, what is diapedesis? Transmigration of leukocytes via endothelial gap is called as diapedesis, right? So this is very important to understand. Transmigration of leukocyte via endothelial gap, not the basement membrane. And which cytokine and which adhesion molecule is most important? That is another frequently asked question in All India exam. So most important adhesion molecule, this is the question. Which is the most important adhesion molecule for diapedesis? This was All India question. Most important adhesion molecule, as I have said just now, is the PCAM1, right? PCAM1, as I have told you, platelet endothelial cell adhesion molecule 1. So that was the answer. PCAM1 is the most important adhesion molecule for diapedesis, right? So this is the older question which examiner had asked many times. Now we will predict what can be the future question, because PCAM1 is an immunoglobulin family molecule. And this immunoglobulin family molecule is also known as CD31. So CD marker is CD31. So that is what we are predicting, that this could be a future MCQ in exam, CD31, right? So be careful, examiner may ask this question. So PCAM1 is also known as CD31. So both are same, right? So most important adhesion molecule for diapedesis is the PCAM1 or CD31. and diapedesis is transmigration of leukocyte via endothelial gap. So now we can see how we can correlate the concepts. When we were studying about endothelial gap, you remember I said endothelial gap will be formed at venule throughout human body except for the lungs, where it will happen in capillaries, right? So examiner had asked question, what will be the site of diapedesis? What will be site of diapedesis in lungs? So that was the question in All India exam 2015. Diapedesis in lungs will occur at venules or capillaries, right? So where it will happen, whatever is happening for endothelial gap, same thing will be happening for diapedesis also, because diapedesis is transmigration via endothelial gap. So in lungs, when they are asking, your answer will be capillaries, not venule, because this is one exception, and that was the answer, capillaries. So diapedesis in lungs will happen at the level of capillaries, right? So there was a good question in AIIMS exam. In AIIMS exam, question was, there was a question, if PCAM1, there was a statement given, PCAM1 will activate endothelium for diapedesis. So is it PCAM1 who is activating endothelium for diapedesis? No. Who is activating? Cytokine. Cytokine is activating. PCAM1 is an adhesion molecule. See this, it's very simple to understand. PCAM1 is a simple adhesion molecule. Simple adhesion molecule which is expressed on both neutrophil and endothelium. So these are adhesion molecule, and who will express this adhesion molecule? Cytokine. So that is why I started like this. If you remember what I have said, during inflammation cytokine will be produced. Cytokine will induce PCAM1 on endothelium, and cytokine will induce PCAM1 on neutrophil also. So both will be because of cytokine. So cytokine will induce PCAM1 on the neutrophil, right? And PCAM1 is not going to activate neutrophil or endothelium. It is cytokine who is activating neutrophil and endothelium for diapedesis, right? So this is the diapedesis. So again, I will continue this blood vessel wall. I will continue this blood vessel wall. So this is the blood vessel wall as I was showing you, right? This is the endothelial gap. This is the basement membrane, and these are the endothelial gap, right? And we were watching, there was neutrophil movement through the diapedesis. This was the neutrophil, diapedesis. So diapedesis is between endothelial gap, and suppose the bacteria is here, right? So now neutrophil has to migrate towards the bacteria. That is why cellular events of acute inflammation is required, right? So now this neutrophil will be moving towards this bacteria, right, because of this chemotactic stimuli given by bacteria. So when neutrophil will be coming outside here and they are moving towards the bacteria, this is called as chemotaxis. So this is the sixth event, chemotaxis. So chemotaxis is the neutrophilic movement towards the chemotactic stimuli, which is bacteria here in this case, right? So the question is, this chemotaxis will be unilateral or bilateral? Will it go there and after killing the bacteria, neutrophil will come back into the blood vessel lumen? It's a bidirectional or unidirectional? That was the question in exam, right? So if you remember, when we have discussed in first chapter, apoptosis, physiological apoptosis is for killing of the cell after their function is completed. Same will be applied for here also. After completion of the function, neutrophil will be killed by physiological apoptosis. So what will be chemotaxis? Chemotaxis will be unidirectional movement. So that is why examiner had asked this question in All India and AIIMS both exam. What is chemotaxis? Chemotaxis is unidirectional movement. Unidirectional movement towards chemotactic stimuli. Towards chemotactic stimuli, right? So that is very, very important to understand. It is unidirectional movement towards chemotactic stimuli is called as chemotaxis, right? So how chemotaxis will be produced? Means what are the substances required for chemotaxis? So that is another important MCQ. What are the chemotactic stimuli? Chemotactic stimuli, right? So chemotactic stimuli, we can divide them into two broader categories. One which is present in our human body, they are called as endogenous chemotactic stimuli. Second, which are from outside, is called as exogenous stimuli, right? So endogenous and exogenous chemotactic stimuli. Exogenous, easy to remember, these are the microbial antigen. These are the microbial antigen, which is exogenous chemotactic stimuli. What are the endogenous within our human body, right? So now question is, what are the endogenous stimuli, right? Endogenous stimuli can be, it can be complement, right? So endogenous stimuli, it can be complement. Which complement is chemotactic? That was all India question, and be careful. Examiner will twist the language like this, and we have to be very careful. What are the chemotactic complement? So remember, C5A is more better than 3A and 4A. So this is the order of the chemotactic ability of the complement. C5A maximum, then 3A followed by 4A. So maximum chemotactic activity is shown by C5A. In All India exam, all were given. So examiner had asked this question earlier, and all will be right answer. C5A, 3A, 4A, all types of complement will be giving chemotactic stimuli. This is number one. Number two, this was the question. Always look for the language, always look for the option. Before you solve the question, you must see all the options. C5A, once you have seen, you should not think this is the best one. Don't think like that. Look at the second option, third option, fourth option will be all of the above. That is why examiner will try to deceive us during exam time. Now coming to the second one. This is another example which was asked in All India exam 2015. Which of the following, see the language, which of the following is chemotactic stimuli for neutrophil formed by arachidonic acid pathway via COX enzyme, right? Via COX pathway. So that is how examiner had asked, when we are talking about arachidonic acid metabolite, arachidonic acid metabolites, right? So we will discuss in inflammatory cytokine. When we will discuss about inflammatory agents, that time we will discuss about this arachidonic acid metabolite. Arachidonic acid metabolites, they follow two pathways, right? One is called lipooxygenase pathway and second is called cyclooxygenase pathway, right? So lipooxygenase and cyclooxygenase pathway. Actually, when examiner had asked earlier in All India exam and AIIMS exam, older time, they had very frequently asked this question, which of the following is chemotactic? Leukotriene B4 is chemotactic. This is the very old and very frequently asked question in exam. Old and frequently asked question in exam, Leukotriene B4. So when examiner had twisted this question in All India exam, via COX pathway, which of the following we will be chemotactic? Many students have committed mistake for this question and they had gone for Leukotriene B4. But we have to understand, examiner is talking about cyclooxygenase pathway. Examiner is talking about cyclooxygenase pathway, and when he is talking about cyclooxygenase pathway, we have to think about prostaglandins, right? Prostaglandin D2 alpha and prostaglandin F2 alpha. So these are the two chemotactic prostaglandin for the neutrophil. So this point also we have to keep in mind. Prostaglandin D2 alpha and F2 alpha, they are chemotactic stimuli for neutrophil movement. So this was the question in All India exam, right? So that point we have to keep in mind. For neutrophilic chemotaxis, prostaglandin D2 alpha and F2 alpha is important. So be careful during exam time. Don't jump to a conclusion because of your earlier questions. Read the language, what examiner is talking about. If examiner is saying lipooxygenase pathway, your answer should be Leukotriene B4, right? But when examiner had asked this question in All India exam was COX pathway. When examiner had asked this question in All India exam was with the COX pathway, then what should be your answer, right? So COX pathway, cyclooxygenase pathway, your answer should be prostaglandin D2 alpha and prostaglandin F2 alpha, right? So these are the important chemotactic stimuli which will be helping in chemotaxis, right? So this is the sixth event. And seventh is when this bacteria will be killed by this. So seventh step is, seventh cellular event of inflammation is phagocytosis. So seventh is phagocytosis. So now we will see the seventh one is the phagocytosis. We all know that phagocytosis will be performed by phagocytic cell, where they will engulf the bacteria or microbes and then they will kill them inside the cell. So these are the seven cellular events of acute inflammation which we have to keep in our mind.